Okay, so you know what's the number of power I must teach right now? So I have told this power for you, and this, and this. And DNA microarray, yes. I told you DNA microarray too. And I told you metagenomics, yes, am I right? I have taught the metagenomics for you too. Please write the number of power for the next section. We have problem every section for starting our lecture, okay? Because I have many classes and I don't remember where I have teach in each class. So you have to write the number. Okay, Metatranscriptomics. Did I teach the Metatranscriptomics for you? Okay, please tell me if I have teach the Metatranscriptomics for you. Okay, no. So what about metagenomics? Did I tell about metagenomics? Here, this power. Did I tell you the metagenomics? Did I talk about Omics Strategies? Okay. So I start from this power, culture independent methods to assess microbial and functional. I think I didn't teach this power, I think. If I am not right, please tell me, okay? More than 99 microorganisms cannot be cultivated under standard laboratory conditions and remain in a viable but non-culturable or VVNC stage. More than 99 microorganisms so cannot be cultured. We talked about this in the past, and less than 1% we can culture. So, development of rapid sequencing technologies and a steady decrease in the cost of sequencing has facilitated the rapid increase in the number of microorganisms identified using gene-based technologies. A variety of molecular methods are available which focus on analysis of nucleic acids, proteins, and lipids from different habitats. So here we have a rapid sequencing technologies that we can just detect the molecules from environment, and then we can analyze them without any culturing bacteria. So with these methods, we can realize and we can know more and more microorganisms, because we don't need any culture media. So these methods are rapid, have a rapid increase in the number of microorganisms. So the number of microorganisms identified using gene-based technologies has been facilitated, and a variety of molecular methods are available focus on analysis of nucleic acids. So we can just take and we can isolate the molecules directly from environment, such as nucleic acids, such as proteins and lipids from different habitats. So we can detect and isolate molecules from environments. This method provides the knowledge of the uncultured world, not just based on analyzing gene sequences pertaining to taxonomic diversity, and also allows studying the full gene complement and metabolic pathways in the community. Various molecular approaches, primarily the Omics Strategies, Metagenomics, Transcriptomics, Metaproteomics, and Metabolomics are being implemented for investigating the environmental and host-associated bacterial communities, and for obtaining more information on their role in the ecosystem, and more specifically their interactions with biotic and abiotic environmental factors. So we have various molecular approaches, such as the Omics Strategies. Do you know what's the Omics Strategies? Do you know what is the Omics Strategies, dear students? I'm waiting for your answers. What is the Omics Strategies? Do you know about the Omics Techniques? The Omics Strategies are strategies that need molecular techniques. The Omics Techniques are contained in Metagenomics, Metatranscriptomics, Metaproteomics, and Metabolomics that are being implemented for investigating for environmental and host-associated microbial communities, and for obtaining more information on their role in the ecosystem. So, in these techniques, we can just detect the molecules from environments. So we can recognize more and more microorganisms from microbial communities, and we can study about their role, and we can guess the role in the ecosystem, and we can realize their interaction with biotic and abiotic environmental factors. So in Omics Techniques, that our molecular techniques, we don't need any culture media and culturing microorganisms, so we can please note, we can detect more than 1% of microorganisms, and we can realize more and more microorganisms. Here you can see about the omics approach for functional microbial diversity analysis. You can see this picture. Okay. Please see here. The title of figure is Omics Approach for Functional Microbial Diversity Analysis. As you see in the picture, a combination of these meta-omics tools can provide temporal information on the uncultivable and complex microbial communities. So here you can see about metagenomics and metatranscriptomics, metaproteomics, and metabolomics as omics tools. You can see here at the top of the picture, metagenomics is realized with nucleic acid or DNA molecules. This technique is based on detection and isolation and recognition of DNA molecules from microorganisms. So we are studying the DNA. In metatranscriptomics method, we are studying the mRNA or messenger RNA. As you see here, we are investigating about the RNA or mRNA. So we call this technique metatranscriptomics. And in metaproteomics, you can see the proteins that are the product of translation of mRNA. So here in this technique, metaproteomics, we just investigate about the proteins here. We detect the proteins, the protein molecules from environment directly. And in the metabolomics, we are studying the metabolites. You can see here many molecules as metabolites, and we can study about their role and their structure. If you have any question, you can ask me. Okay. Zahed Kavan. Did you understand the omics techniques, or if you need, I can repeat that. Okay, now we repeat the omics techniques or omics strategies, such as metagenomics, metatranscriptomics, metaproteomics, and metabolomics. They are molecular approaches that we just detect the molecules from environments and then investigate that molecules, such as nucleic acids, such as proteins, such as mRNA or another. So we don't need any culturing of bacteria, we don't need any culture media, and so We call these techniques Omics techniques. Okay, and then metagenomics is a culture-independent method for elucidating the genomic components of a microbial community in a common environment. The first technique that we are going to study about the omics techniques are metagenomics, as the title. Metagenomics is the first technique we are going to study, the first technique of omics techniques. Metagenomics is a culture-independent method. As another omics techniques, this method is a culture-independent method too. So we, as I told you before, we don't need any culture media. Mr. Zahed Kavan, this strategy is for recognizing the bacteria and for the taxonomy of the bacteria. As I told you before, over the 99% of microorganisms are V, B, and C. They are not culturable, so we cannot detect and recognize them. So we use these techniques to detect them. Why? Because we don't need any culturing media or culturing of microorganisms. Just we detect the molecules from environment, such as nucleic acid, protein, or another molecules. So we can realize, we can guess more and more microbial communities. More than 1% of bacteria can be recognized. Okay, and then it has been termed environmental. You're welcome. It has also been termed environmental genomics and community. In genomics, it does not require the presence of cultured organisms, just viable ones. So we name this technique environmental genomics 2 and in community genomics 2. It does not require any culturing of microorganisms, as I told you. It's primarily based on shotgun sequencing and results in two types of profiling of whole microbial community: taxonomic profiling and functional profiling. Taxonomic profiling focuses on characterization of the presence, abundance, and evolutionary relatedness of organisms present in the microbial community, while functional profiling is concerned with the organism's behavior in communities and ecosystems. So here we have two types of sequencing techniques. One taxonomic profiling and another functional profiling. So we investigate in two aspects: taxonomic profiling and functional profiling. Taxonomic profiling is investigating the characterization, abundance, and evolutionary relatedness of microorganisms. But functional profiling investigates the behaviors and the traits of microorganisms in the ecosystem. If you have any questions, I'm waiting for you. Okay, if you don't have any question, I can continue. Metagenomics involves the genomic analysis of unculturable microorganisms by direct extraction and further cloning of DNA from the microbial population of a particular habitat. So metagenomics is, as I told you before, the genomic analysis. It analyzes the genome of bacteria that we cannot culture them. So it is a genomic analysis of unculturable microorganisms. We extract directly the DNA and then we can investigate the genome or libraries of microbial communities that we have provided. And they have many useful information for us for investigating the functional and the capabilities of microorganisms. So metagenomics libraries of microbial communities provide vital information about the phyla present in the particular habitat and their functional capabilities. Okay, going to make it bigger for you to see. Here, this picture is metagenomic library constructions and functional annotation. So here you can see the processes in metagenomic techniques. You can see here, as I told you before, we have to isolate the samples. We have to screening, okay, screening the environment. So we can use the environmental samples such as soil, such as water, such as rumen, and such as human microbiome. Okay, I'm going to get it bigger for you to see. So here we can use many different samples from many different environments. Soil, water, rumen, human microbiome. And then the next step is to isolate the DNA. So DNA isolation is the next step. And the third step is creation of clone library containing DNA. When we have isolated the DNA, we can create the clone library. And the last process is extraction of metagenomic DNA in heterologous host. So we use this clone library and we insert them to bacterial cells or host cells. So at the end, we can see the role and the functional microorganisms, functional screening of metagenomic library. When the genes are expressed, we can see the ability of microorganisms here at the end. If you have any question about this picture, you can ask me. In this picture, you can see all of the steps for metagenomics techniques. Dear students, do you have any question about this picture or not? So the first step is isolating the samples, then isolating the DNA, creation of clone library containing expression of metagenomic DNA in heterologous hosts. So if you don't have any question, I'm going to the next part for you. There is an increasing demand for chemical production of novel enzymes and biomolecules that are required for large-scale industrial processes. As you know, today is an increasing demand for some enzymes and some chemical enzymes, and we are looking for novel enzymes too, so this technique can be useful. Metagenomics helps in identifying novel microbial populations from complex environments such as soil, ocean, groundwater, cow, rumen, and the human microbiome. So as I told you before, many environments can be screened. We can screen many environments for looking for new and novel microorganisms, novel enzymes. When we find new microorganisms, so we can find new enzymes too. These organisms are expected to produce biomolecules with enhanced activity profiles that can be further utilized at a larger scale in bioproduct-based industries. So these organisms can produce biomolecules or enzymes with new activity, and we can see for more and more new enzymes and novelities as bioproducts. We can use them in industries. Along with the identification of novel biocatalytes, new antibiotics and bioremediation are other fields with a demand for new bioproducts. So as you see, many aims and many products are important too, such as biocatalytes, such as new antibiotics that we are looking for them, and they are very necessary for our life. And bioremediation, role of bacteria are more important than in the past. So we are looking for novel activities from microorganisms in our life, and we can recognize them with metagenomics or omics techniques. Because of the technical advances in metagenomics, mining of enzymatic activities from the microbial community of different habitats, many of the industrially important enzymes like cellulases, lipases, gazellanases, amylases, and proteases are intended to be produced at a large scale using metagenomics. So as we are looking for novel enzymes, as I told you before, many enzymes can be used in different industries, such as cellulose, such as lipases, such as gazellanases, such as amylase, and such as protease. They are important enzymes that we can use them in different industries. So we are looking for new enzymes with new conditions, with more production, and etc. with metagenomics techniques. Functionally, analysis of metagenomic data have produced information about new antibiotics, hydrolytic and degradative enzymes, biosynthetic functions, and antibiotic resistance enzymes. So, as I told you before, functional analysis of metagenomic data are very important for us too, for getting information about producing new antibiotics or new hydrolytic enzymes or degradative enzymes that are very important for degrade new materials or producing new antibiotics, or biosynthetic functions, and for antibiotic resistance enzymes. Okay, the next technique is Metatranscriptomics. If you have any question until now, you can ask me, or if you don't have any question, I can continue. Okay, metatranscriptomics is another technique. Metatranscriptomic analyzes the study of the RNA complement from the entire microbial community, determines which microbes are active and which genes are transcribed. So metatranscriptomics analysis is the study of RNA complement, as you see. As I told you in the picture, we have to study the RNA complement, as I told you in this picture. I'm going to return to that picture. Here. In this picture, as I told you before, we are going to study the RNA or mRNA. That the technique is metatranscriptomics. We are going to study the transcriptome in this technique, that the transcriptome is equal to mRNA or RNA. Okay, so metatranscriptomics analysis is the study of RNA complement from the entire microbial community, determines which microbes are active and which genes are transcribed. So in this technique, we are going to study the RNA complement and the mRNA or messenger RNA. For true analysis of the transcriptome, metatranscriptomics and metaproteomics are the new evolving disciplines within the metagenomic field. The total RNA in a microbial community is sequence and analysis. So we are going to detect the total RNA from environment. We just have to detect the RNA molecules from environment directly, and then we are going to recognize the organisms, the microorganisms. So the RNA can be detected from environment directly. RNA is extracted from the sample and complementary cDNA synthesized. Okay, why we are going to synthesize cDNA? Do you know? Are you present really in the class? So if you are present in the class, you can answer my question. Why we are going to study to synthesize the cDNA molecule from RNA immediately? Do you know why? Okay, I asked you why we synthesize the cDNA molecule from the RNA. Why we synthesize the RNA, the cDNA molecule immediately from RNA. When we isolate the RNA from environment, we synthesize the cDNA molecule from that, from mRNA, from RNA or mRNA. We change the RNA molecule to cDNA. Okay? Why? Why we have to synthesize the cDNA from RNA? I think some of the people and some of the students are not really present in the class, and only their name is present and are shown in the class, but they are not really in the class. They are doing another works. Okay, so the reason that we have to synthesize cDNA from RNA is because the RNA molecule is not stable. It is degradable, and it will be degraded after 10 minutes in the environment. So the molecule, the RNA molecule is not constant, it's not stable, and we have to change it to cDNA molecule immediately for the stability. So here you can see RNA is extracted from the sample and complementary cDNA is synthesized. For the functional profile analysis, the RNA reads of functional genes after sequencing are mapped and functionally annotated. So yes, I know, Mr. Jabber Abi, thank you. We use the RT enzyme for converting the RNA to DNA molecule, but I asked you why. The reason of why is that the RNA molecule is not stable, but the method, yes, you're right. Thank you. cDNA can then serve as template in a variety of downstream applications for RNA studies, such as the gene expression. Therefore, cDNA synthesis is the first step to many protocols in molecular biology. Yes, you're right, but I'd ask you why, and you must answer the reason of why, not how. Okay, you told how we can synthesize cDNA from RNA, but I asked you why we are going to synthesize cDNA from RNA, because the RNA molecule is not stable. And thank you. Okay, in this picture you can see the steps of meta-transcriptomics analysis. First of all, we have sample collection. The next step is RNA extraction, and the third step is cDNA synthesis. This, as I told you before, meta-transcriptomics sequencing is another step. As I told you, we are going to sequence the cDNA, and the mapping of RNA reads are another steps, and the final functional annotation is another, and the last step. So we can investigate and we can recognize the function of that molecule RNA. And we can investigate the role of that microorganism. Environmental metatranscriptomics focuses on isolating and sequencing mRNA from microbial ecosystems to assess the genes that are expressed in that community. So we can recognize the genes that are participating in a special activity or a special role in ecosystems. So such studies have been used to analyze samples from different habitats, such as water, soil, sediments, seawater, gut microbiome, and activated sludge environments. Metatranscriptomics has also been used for the assessment of microbiome immune interaction, supplementing 16S rDNA data. So here you can see we can use this technique for analyzing the samples from different environments, such as water, such as soil, such as sediment, seawater, and gut microbiome, and also sludge environments. And we can use metatranscriptomics techniques for assessing of microbial immune interactions, and that is very important too for investigating the immune interactions. Okay, your teaching is stopped here because of the Ramadan. I'm going to teach you shorter, and I'm because of your demand of many students, I have shortened the time of the class, and your lesson is not behind. So your lesson is until Metagenomics. Please write the number of the power 30. And until the power 30, please, the representative or a person of your class, I don't know who is your representative, please write the number of the power. Every section we have this problem for starting the section. So please write the number of power 30, and in the beginning of the next lecture, when I ask you which power I have to teach you, you please tell me number 30. Okay, so immediately I can start the power and I can continue the lecture. But we have to lose, and we miss the class time. Yes, I will send this lecture to your WhatsApp in as soon as possible. I don't know when, but I have to. Yes, I will do it. And if you have any questions, you can ask me. And if you don't have any questions, I finish this lecture, and for another section we have to talk about metaprotomics, protogenomics, and about advantages and drawbacks of these techniques, and microbial evolution. We are going to talk about that in the next lecture, and microbial phylogeny too. Okay. If you don't have any question, I hope for you a good day and a nice week. Take care of yourselves, and goodbye.